Welcome to our presentation on the paper Alzheimer's disease diagnosis via Deep Factorization Machine Models. Alzheimer's disease is a slowly progressing neurodegenerative disease which is highly heterogeneous and not yet fully understood. It can be divided into three states of progression: cognitive normal, mild cognitive impairment, where patients are not yet affected in everyday life, and Alzheimer's disease. A reliable diagnosis of these three groups is highly important for future research. We use biological markers for this task. Biomarkers for Alzheimer's are present in multiple modalities like demographics, magnetic resonance imaging, single nucleotide polymorphisms, and biomarkers extracted from cerebrospinal fluid. The goal of our paper is therefore a good performance for the diagnosis of all three disease stages by using biomarkers and, importantly, the interactions between biomarkers of different modalities. In addition, we aim to have a high interpretability of the results. This is achieved by combining the interpretability of linear models with the performance of neural networks. Linear models, in our case factorization machines, model pairwise feature interactions explicitly, while neural networks model high-order interactions implicitly. Our model structure is therefore similar to DeepFM and looks as follows. The multimodality input features are fed into an embedding layer which embeds features of different lengths into dense embedding vectors with a set length. Then our model incorporates a factorization machine. First, this factorization machine gets the raw features to combine linearly. Second, it computes pairwise interactions between all embedding vectors. Lastly, we implement a neural network which computes high-order interactions from the concatenated embedding layer. Factorization machine and neural network outputs are then combined. Furthermore, the embedding layer enables us to use meta-embedding. This means combining multiple features into one embedding vector. We only combine volumes of brain parts into larger regions as this is medically reasonable. For example, the parts of the parietal lobe together. This will later also help with the interpretation of results. The data comes from the Alzheimer's Disease Neuroimaging Initiative, has patients of all three disease stages, and incorporates all of the aforementioned modalities. For training, we use five-fold cross-validation. After training, our model performs as follows. Our model outperforms the other models with a median balanced accuracy of 0.589. This shows that the combination of factorization machine and neural network performs better than each of the models alone. The linear interaction modeling, however, is unstable, which underlines the capabilities of factorization machines. Using the previously explained meta-embedding, the model exhibits even less variance than the original one. As the goal of our paper is also the interpretability, we examined the features that are most important for a prediction. The most important features are extracted from the linear part of the factorization machine. On the right, the top 10 most important features and their importance is displayed. All of the features are medically meaningful. The two most important features, for example, are the hippocampus and the entorhinal cortex. Both are known to exhibit atrophy due to Alzheimer's disease. In addition, even the signs of the features match the disease pattern. For example, the larger the hippocampus, the less likely the patient is to have Alzheimer's. Furthermore, our model design enables us to not only extract single feature importances, but also feature interactions. Unfortunately, due to the large number of features, an even larger number of interactions contribute to the result. One can only identify that many regions in or near the hippocampus, as well as a few genetic markers, appear as important. However, an interpretation is difficult. At this point, meta-embedding helps again. It leads to less features and therefore more interpretable results. Our model with meta-embedding especially utilizes the temporal and frontal lobes. This shows that overall our model not only outperforms compared models, but also enables a thorough analysis of the most important features and feature interactions. Thank you very much for your attention.